QIMR Berghofer acknowledges the traditional and cultural custodians of the lands, waters and seas across Queensland, and we pay our respects to elders past and present. We support the cultural knowledge, determination and commitment of First Nations communities in caring for health and well-being for millennia.
You picked this podcast, so you probably already know metastatic triple negative breast cancer is the most aggressive form of breast cancer, affecting women, mainly young women. It's incurable and researchers urgently need answers. And now, because of a very unique collaboration, a real opportunity for these answers exists. Thanks to QIMR Berghofer's Professor Sudha Rao and the CEO of Kazia Therapeutics, John Friend, who both join me now.
This is BodyLab, a QIMR Berghofer podcast.
Brilliant to have you in the studio, Sudha. You're very hard to nail down. You're a very, very busy woman, and it's great to have you and also your collaborator, John Friend, CEO of Kazia, in the podcast suite, and we can talk about this really exciting research. I know for you, this is personal.
Really, I guess for most of us, we have been touched by people who've had cancer, and I'm no different. And I think my journey really started in 2015 when a very close friend of mine, Melanie, she was only 39 years old, and she just had given birth to a second child, and she found out that she had stage 4 metastatic breast cancer. I went through that whole journey with her and realized at that time that the only treatments that were there for her were chemotherapy and radiotherapy and so forth. And I remember her calling me and saying, my cancer has gone to the liver, and it had metastasized to the liver. Within 10 days, she passed away. I realized through that journey that we have to do something in terms of helping patients with metastatic breast cancer. And most recently, only about 2 weeks ago, my own father passed away from metastatic cancer as well. So I know more than ever now that, we have to have treatments. And he always said to me that, whatever you're doing, keep going. And so it's become even more personal for me in terms of how we tackle it. I think it's through Melanie's experience that I realized that as scientists, we talk about clinical research, we talk about translational research, and here I was as a researcher going, okay, what can we do? And one of the key aspects that we only touched upon with Melanie was that we realized that if we're going to tackle metastatic cancers and metastatic breast cancer, so we have to get the patient's own immune system to fire up. And if the patient's own immune system can actually kill the cancer, then we're moving forward. And that's really what immunotherapy is. Immunotherapy is about harnessing and allowing the patient's own immune system to work. What the problem for cancers like breast cancer, like for example, triple negative breast cancer is the most aggressive form of breast cancer, is immunogenic, means that immunotherapy works, but it only works on a subset of patients. What we wanted to do with our work is we wanted to identify mechanisms to be able to allow immunotherapy to be available to all breast cancer patients. And that's really where we started our journey about trying to understand the cell types that are important for metastatic breast cancer. And that's when we realized, together with other labs, that really the metastases or the spread happens because of these dormant cancer cells, which standard of care doesn't treat. And so these dormant cells, then after standard of care, they move into niches and they may stay there for a while, for weeks or months. They spread to other parts of the body. And when they do, that's when the spread happens. So we realized, can we take these dormant cancer cells and can we peel them apart and start to understand the molecular signatures underneath that? And if we can understand that, then we can actually make those cancer cells immune visible and then allow immunotherapy to work. And so fast tracking forward, that's what we identified in this collaboration, was that when we started to peel apart these breast cancer cells, we identified that there was a unique PI3 kinase epigenetic switch, which is harbored by these dormant cancer cells. And we realized that using various methodologies, that if we can block this PI3K epigenetic switch, then the cancer cells became invisible to the immune system, and then the immunotherapy started to function. And so we needed a drug to be able to do that, because we identified that in a test tube. And that's when we contacted Kazia, who have the drug Paxasilib, which actually targets the PI3 kinase pathway. But they had not, or no one has shown that it can work within the nucleus as an epigenetic switch. So when we got their drug and we started to put it into what we call preclinical animal models, which are immunotherapy resistant, so immunotherapy doesn't work on them, what we found was by adding the drug Paxasilib together with immunotherapy, we suddenly realized that not only the primary tumor was completely abolished, but we also prevented the metastases from happening. And that was for us an enormous light bulb moment, because here we had an animal model which was completely resistant to immunotherapy, and we had prevented not only the primary tumor burden, but we also prevented, importantly, the metastases, the spread. And we also then went on to show that in patient-derived blood samples and so forth, and showed that by using this drug together with immunotherapy, we were able to kill these dormant aggressive cancer cells. So this is fantastic, because it opens up the whole idea of being able to treat metastatic cancers. And it's something that I'd obviously promised Melanie many years ago, that that's what we would do, is to harness immunotherapy. So that's how this whole journey started.
And you talked about epigenetics, so can we just clarify that for people who haven't heard it? This is still an emerging field, although you've been doing it and your team for quite a while. What does it mean?
So epigenetics, to me, is incredibly exciting and very unique. So epigenetics is really what I consider is the layer above our DNA sequence. So it's independent of any gene mutations. And it's that layer that really responds very elegantly to environmental signals. It dictates what genes are being expressed or not expressed, what proteins are being made in the cell. It's what I consider very much the instructions manual of a cell. And what's really unique or exciting about epigenetics is really twofold. One is that epigenetics is reversible. By reversible, it means that we can rewrite the instructions manual of a cancer cell to make it look more like a healthy cell. It also has what we call memory. So well after all the proteins and the RNA, etc. is gone, the epigenetic footprint from a drug is left behind. And if it's left behind, it means it has huge implications in dosing, because if the treatment is working, you don't need to keep dosing the patients for a long time. So it means that it has implications in safety and toxicity and so forth.
I know in your research, you found that actually lower doses worked better.
That's a very important point, because that's another aspect that's very exciting here, is because this opens up the idea is, can other drugs also be used in a different way? So what we actually found was that by lowering the dose of the drug, that's when we allowed the cancer stem cells, or these aggressive metastatic initiating tumor cells, to be immune visible. And that's when the immune reinvigoration happens as well. So this is quite contradictory, I guess, to what people think, that normally we'll use higher and higher doses, and it's almost like a sledgehammer approach. But what we believe is that by lowering the doses, the more selective mechanisms can work.
It's extraordinary that you had all these things, and they didn't magically come together. There's tireless work behind it. So how does a scientist find the perfect drug like this, John?
I don't know about perfect drug, but I appreciate the offer.
Perfect for this scenario.
For this, right. So as you heard Sudha really describe, and the excitement in her voice, as well as what I hope you hear, the excitement in my voice about this collaboration and potential, the way that she went about this and the way her team went about this was really to understand the mechanism of action, how this epigenetic sequencing occurred, and also the targeted PI3K aspect within the DNA. From there, it was a matter of, finding out what asset or what potential drugs could be applied in some of our preclinical studies, but more importantly, what then could be translated into a clinical study into humans. There are several PI3K inhibitors out there in the clinic for lymphoma populations, but also for breast cancer populations. What was unique about our molecule, paxalisib, is that not only is it hitting that PI3K sequence, but it also has been shown that it can cross the blood-brain barrier. This is extremely important for a lot of tumor types, especially in breast cancer, triple-negative breast, but also other tumor types like non-small cell colorectal, whereby patients have metastases. They metastasize not just to liver and bone, but one of the more frequent metastatic sites is the brain. And we know that once metastases occur in the brain, progression is quite advanced and quite quick. Paxalisib was originally designed to cross the blood-brain barrier. That was one of the key features when they discovered this molecule. It was designed and discovered specifically to be able to cross, and therefore our focus for the past few years has been in brain cancer. It's been in both adult and pediatric brain cancer. So when Sudha found me, it may have been an email that came across my desk, and I thought it was quite unique. And coming from, being an Australian-based company, of course, I reached out and we hit it off immediately. I mean, we had a call, and her hypothesis, right? Because this was a concept. It was a hypothesis of this potential benefits of a PI3K inhibitor in conjunction with immunotherapy to really advance the treatment of breast cancer. And there's a huge unmet need, as Sudha mentioned, that for patients that this immunotherapies work, it works really well. But that's only a small percent. 30-40% really see this sort of impact and benefits of immunotherapies. The rest see zero effect, zero benefit.
It's heartbreaking.
It is. And the potential that this could be advanced from a concept to in vitro cell line testing to in vivo animal models, and then potentially to the clinic was one thing that I found extremely exciting. This is novel science. Do your Google research. You're not going to find this sort of research out there because it's novel. That's also what I found extremely intriguing and exciting to jump into this collaboration. Obviously, QIMR speaks for itself in terms of their research, and in a very short period of time, the cell line, the in vitro, the in vivo testing, and we've even gone well beyond that, including some sequencing, some biomarker work. So it has accelerated such that in the 2 years, I believe, we've been collaborating now, we've probably done what most academic institutions and biopharmaceutical collaborations would take 5-6 years. From the time we spoke until we signed the agreement and then initiated work, it was only a few months at most, maybe 2 months at most.
How did you become aware of this drug, Sudha?
As John has mentioned, we have always been interested in the triple-negative breast cancer that goes to the brain, that spreads to the brain. Unfortunately, most young women, the metastases will go to the brain. We were aware of paxalisib because we'd identified this novel mechanism in these dormant cancer cells, and we knew that switch was, that PI3K mTOR switch was important. That's what we do as scientists. You just have to try. We knew about this drug just through the literature and so forth. I just went up, looked up on the website, looked at sort of contact details, and I wrote an email, and that's how the whole, we never looked back. I mean, when you have a fantastic partnership, I feel like I've known John all his life. We say to each other, oh, is it just been 2 years? When you click that way and you have a common goal, and I think when we say that we have a common goal, it's because at the end of the day, both of us have been touched by family members or friends who've had this disease. Both of us have the same goal. We want to change patients' lives. We want to give patients more time. Fundamentally, that's what drives us both, and we've often talked about that. And I think that's the reason why we don't really care about the nitty-gritties of whatever. We just, if I need something, John's gone, okay, let's see what can we do. If John needed something, I've said, okay, let's see what we can do. And so we've just made it happen, because if we can help another couple of patients, that's our goal. And I think that's fundamentally, I think that's where we both come from.
I don't know if you know this story about Sudha. Her story with Melanie taught her a big lesson. I should let you tell this. And it was about asking for funding. Do you know what I'm talking
That's
about?
absolutely. So at the time when I knew Melanie, at that time I was desperately trying to get funding to be able to do breast cancer research. I used to say to Melanie, "Oh, I'm worried. What can we do?" And she said, "Why are you worried? Why are you worried what people say? If you don't ask, you will never get." And so she taught me that the way to sort of go, well, actually, you have to ask if you want to move this forward. And I'll always remember that. And I always remember Melanie every day still. She's with me all the time because there's at the back of me somewhere along the line, she's always pushing me. It's exactly that. Thanks for reminding me, because that's exactly what this is all about.
It is, and it's where you come in again, John, now, because I
Yes.
know with Daniela, can you tell a little of that story, Sudha?
Yeah, so we had paxalisib already, and I got a phone call from Daniela's family. We're sort of a lab that's known to be able to test people's bloods and so forth. And they sort of basically told us that the daughter, she was only 28, I think it is, and she had glioblastoma. And I actually talked to Daniela. And when I was talking to Daniela, she was like, "Yeah, I think I've got something, but I'm fine." She was really, into all her sports and stuff. And then she started to deteriorate very quickly. I mentioned to them, we're using this drug called Paxasilib, but I said we're using it for a completely different indication. I'm not an oncologist, etc., but maybe you want to mention it to the oncologist. Before long, then they contacted Kazia, independent of me, because I'm not an oncologist. And Kazia, through their goodwill for compassionate purposes, did everything in their capacity to get the drug here for Daniela to have it. That just speaks again for how John is. He just took whatever he could to get it here. But unfortunately, that was too late for Daniela. But she did take a few doses of the drug. And when she did take them, we did sample her blood. We found incredibly that her immune cells had started to reinvigorate. This was for us a game-changing moment, a goosebump moment, because we sort of thought, okay, let's just test the bloods that the oncologists importantly had provided us. And we said, okay, let's just test to see what happens. This is in glioblastoma, we don't know. But my lab is known for looking at the exhausted T cells. By exhausted T cells, I mean these T cells can't kill. So we took the bloods and we thought, let's just make the most of it, because at least we can tell the family if there's any evidence of anything happening. And that's when we first saw that the T cells had been reprogrammed and they had killing function. And so that's the part that for Daniela's family, they're big advocates of this drug because of Paxasilib, because even though it was unfortunately too late for Daniela, the fact that you could see that through Daniela's blood that those immune cells had been reinvigorated, and this could then have potential for other patients in the future, for them was a really important moment.
Big breakthrough for you, John.
Very much so. It was just another component of this collaboration, and also just complementary to the initial PI3K sequencing, epigenetic resequencing, that not only are we showing the sort of dormant cancer cells becoming reprogrammed and visible to our own immune system, but also reinvigorating our own immune system. This is all novel science that, we had no idea that a PI3K inhibitor could do. In terms of compassionate use, expanded access, that's something I've always been very passionate about, and that is providing for patients and family members compassionate use when there's really nothing else, that there are no trials. Unfortunately, that there were no trials available at the time for Daniela, and we make every effort that we can, being a small biotech company, to potentially have that benefit for those patients.
You have a great team, Sudha, but you really have those two women still part of your team.
Absolutely. No, absolutely. I mean, I think when I say we have both those women as part of our team, I cannot underestimate that my whole laboratory, my whole team of researchers in my lab, many of the researchers in my lab are very young, early career researchers, mid-career researchers. For those guys to be able to see the benefit they are making to patients, I don't need to convince them to come at 10 at night or come at 6 in the morning, because they know that they're making a difference. So that's why I think that those two women will always be in our lab members, because that's what drives a lot of the people in my lab, because they see that their bits of their research is actually helping patients. And I think that's really important.
This is an extraordinary pairing. We've worked that out. So where to next?
I think from my standpoint, the next steps is this translation, right? We always talk about translational research. And
What does that mean to a layperson?
yeah, from my standpoint, it's essentially taking bench work, lab work, and being able to then convert it into a clinical study. And that's essentially where we're at now, is taking this research and translating it into a clinical study in advanced breast cancer. So we are and have worked very closely together, as you can imagine, in designing a clinical trial in Queensland, right now focused on 3 clinical sites and major world-renowned cancer centers here in Queensland to launch a clinical trial. We'll be enrolling 24 to 26 advanced breast cancer patients, including triple-negative breast.
That's a 1B?
This would be a phase 1B clinical study. Again, the collaboration is still remaining within QIMR. QIMR and Sudha's lab is going to be supporting in terms of biomarker, liquid biopsies, and there's even more in terms of this collaboration that we've also expanded, going even potentially beyond advanced breast cancer.
Something else that is a part of this that we've only touched on, but I'd like to know more about, you can find out in real time how this drug is working. Instead of being on the drug for 4 to 6 months and then seeing what the result is, you developed a liquid biopsy that is able to monitor in real time as we go along.
Absolutely. So this is, again, coming back to what I started with Melanie, really, is the importance for being able to track the progression of the cancer.
In a non-invasive way.
In a non-invasive way. So the idea is that we can detect these aggressive dormant cancer cells, but also the immune cells in the bloods in real time in a non-invasive way. And what we're doing as part of this trial, which is pretty novel, is that we're not only doing what is a phase 1B trial, that is, we're trying to understand the safety and so forth, but in real time, we were able to sample the bloods to not only see if the drug is working in terms of whether it's hitting the target, but also importantly, whether it's reinvigorating the immune cells. Is it starting to reinvigorate the immune cells? Are the cancer cells now becoming less aggressive? Are they now being killed off? So we're getting a real-time map of how the patient's cancer is progressing with the trial.
It's extraordinary. I know you've both been very good today putting this in lay terms for all of us who are listening. Well, the stars didn't align, you made them align. So you needed the right team with experience in emerging area of research, which is epigenetics. You needed the right place with the right support and technology, QIMR Berghofer. You needed to make the dormant cells visible. Mm-hmm. You needed the right agent to rearm the immune system, the ability to monitor the results in real time with the blood test, and now you need the right people for the clinical trial.
Absolutely.
And John, you're really putting your money where the mouth is. Not only did you fund the collaboration that's happening on here, your company is funding a clinical trial and funding for any researcher. Sudha's nodding. Mm. It's the beginning and the end.
As you said, we are putting our money where our mouth is. I believe in this collaboration. I believed in it when I first met Sudha, and we started talking about this potential hypothesis and converting from in vitro to in vivo, and then this clinical study. So we are fully behind it. We are launching this trial in 24 advanced breast cancer patients in Queensland with 3 centers. The trial will be in triple-negative breast and advanced breast cancer patients. We're looking forward to this, as well as even expanding our collaboration within QIMR to look at this asset and this mechanism of action in other tumor types, including non-small cell lung cancer and colorectal and other tumor types.
I can't wait to see where this goes. And the both of you, given everything you do, has truncated this process from how long, Sudha, to how long?
So normally this would take anywhere from 5 years to 8 years or even 10 years. And we've truncated all of that to 2 years. And that to me is just amazing in terms of helping patients.
The biggest thing for patients is time, isn't
Exactly.
it? If you'd like to know more about Professor Sudha Rao, her team and their work, QIMRB.edu.au. John and Sudha, thank you both so much.
Thank you.
Thank you.